You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Dr. Florencia McAllister. She works at University of Texas, MD Anderson, Department of Clinical Cancer Prevention, Division of Cancer Prevention and Population Sciences. So, Florencia, thank you for coming. How are you doing?
Yes, hi. How are you? I'm doing well, thank you.
Good. Well, please tell me about your work at MD Anderson.
Yes, of course. So I focus on seeing patients with pancreatic cancer and also patients at high risk for pancreatic cancer. We work on screening these populations. Our long-term goal is to develop better preventive and therapeutic strategies for pancreatic cancer. So if you want, I can tell you a little bit more about the work that we've recently done.
Well, tell me a little bit about pancreatic cancer. I guess is a very deadly, low survival rate form of cancer. Can you just give a few words on the seriousness of it?
Yes, of course. So it's a very, very aggressive malignancy, and despite all the knowledge that we have about the mechanisms and the physiopathology, the progress in therapy has really been very slow. So the survival rate still below the 2 digits. It's around 9% in 5 years for patients diagnosed with pancreatic cancer.
Right. So what are you working on in particular? What's your thinking and research about in regards to it?
Yeah, so mainly I've been very interested on understanding sort of what is the interaction of the immune system with with cancer from the very early stages, trying to understand why at some point cancer can emerge and cannot be visible to the immune system, and how to sort of target that and kind of change that process. So that has been kind of the focus of our lab, and we are very curious about all the environmental changes that happen around tumor formation from an environmental immune perspective and from a microbial perspective.
Yeah, I saw that in the description. Do tumors have their own microbiome, or do they attract specific bacteria?
Yeah, exactly. Yeah, so we actually started looking into that after we published a few years ago that there's a type of T cell called Th17 cells, which actually play an important role in tumor initiation. And we actually asked the question, why do we have those T cells in the tumors in the first place? The real job of those cells in the body is to fight infections. So we thought, why would we have those cells in the first place? So that was really the motivation for us to look at the presence of bacteria, to understand again what triggers the presence of those cells in the tumors. And indeed, we found the presence of bacteria in tumors in murine tumors and also in human tumors. So that was around the time that Dr. Strassman at the Weizmann Institute described the presence of bacteria within pancreatic tumors. So that was really what made us very interested about understanding further on those bacteria in tumors.
Will you characterize the bacteria on tumors a microbiome of the tumor, or just one or two strains or species that happen to appear on tumors?
Yeah, so we focus on the microbiome, on all the microorganisms, because again, when Ravid described the presence of bacteria, there were some species, some genuses that were predominant, but we really wanted to understand is there any role for these bacteria, or at least is there any association of some bacteria with better outcomes? And that's what we decided to do. We basically use 16S to amplify DNA of bacteria species, and with the idea of further deeper characterize what are those species present on tumors.
Well, doesn't that seem to indicate that the pancreas itself has its own microbiome? Has that been studied?
Yes, exactly, and that's what we think. Definitely, the pancreas has its own microbiome, and it's likely different from the microbiome in tumors, as we actually shown in our study. We looked basically on patients that were going to go to surgery at the microbiome in the stool of the patients, of the tumors, and then the adjacent area, which is never completely normal, but sort of the area that is surrounding the tumor that doesn't have dysplasia. And we found the characteristic microbiome in those three areas of the same patient. But what was really interesting is that there was around a 20% overlap between the bacteria found in the tumors and bacteria coming from the gut, whereas those bacteria were absent in the normal adjacent region. So it was suggesting that there's a specific sort of axis or communication between the gut and the tumor. So that was one of the very interesting things that we found.
Well, to my knowledge, there's a couple of different kinds, at least, of pancreatic cancer. So the tumors tend to form in certain areas of the pancreas, depending on the kind, or is it randomly anywhere in the pancreas they form?
Yeah, well, I mean, the most frequent are in the head, but there are also tumors in the body and tumors in the tail. And, we have not really focused on that, but I know of other groups who have been interested on understanding if there are microbial differences that will dictate where the tumor will locate, which I think it will kind of make sense because of the different flow dynamics, etc. But yes, that's something very interesting.
Well, what if every different cell type in the pancreas and in the body has its own unique constituent microbiome? Or do you think it only occurs perhaps at the organ level? I mean, you're seeing it at the tumor level, it looks like, and a tumor is, I don't know, a few billion cells. I don't know how heterogeneous they are, but what are your thoughts? How far down does this go? How specialized are microbial colonies to their
Yeah, no, I mean, those are great questions. I mean, in our experience, when we did look at the tumor microbiome and compared patients that had tumors at different locations, we did not find differences or signatures of tumor microbiome depending on the location. Although I have to say, the number of patients with like tail, for instance, location was much lower. So we didn't design a study that had power to detect differences based on the location. But those studies are kind of easier to be done in animals because you can actually ramp up the numbers of tumors accordingly. But yeah, no, those are good points. We haven't looked in detail at that. Yeah.
Are you getting the samples just from the fecal matter, or when you resect parts of the pancreas or open up the animal itself, are you literally seeing and culturing bacteria that are, in situ at that moment, if possible?
Yeah, that's exactly what we've done. So from the patients, as I mentioned before, we compare the stools with resected tumor and resected adjacent areas. And from the mice, of course, since we have the flexibility of obtaining more tissue, we usually, we will look at the stools and then certainly, the tumor and then different areas within the pancreas and then also different areas of the duodenum or, small intestines. Yeah.
So what have you noticed about the microbial constituents of tumors how do they differ from the overall pancreas microbiome?
Yeah, so the study we designed is we thought if tumors microbiome are important and relevant, and if we'll have to look at some association start by looking at association between certain bacteria and sort of tumor, like patients' outcomes. So unlike other, tumor types in which you can actually, compare patients that, do respond to certain therapies versus those who don't, in pancreatic cancer, that's much harder to do. So we thought we can actually look at the patients that do survive long term, which although they are very few, they are, 1 in like 50 or so. And by long term, I mean patients who live more than 5 years after resection, with an average of like 10 years. For pancreatic cancer, that's really considered long term. And we compare them with matched control patients who live less than 5 years with an average of 1 year and a half after resection. All these patients, of course, went for surgery, so they had early disease diagnosis, either stage 1 or 2. So we compare those two groups of patients, and because we knew this was a kind of new area of research, we wanted to have a second cohort to sort of validate these findings. So we had a second cohort. Our first cohort, the discovery cohort, was from our institution, from MD Anderson, but then we were very lucky to keep in contact with Johns Hopkins group, where I did my fellowship training. And, through Laura Wood, there we were able to access also a cohort from Hopkins of long-term survivors and short-term survivors. And what we found basically is that in both cohorts, the tumor diversity in patients that lived long-term was much higher than those patients that lived short-term. So basically, the diversity, or also called alpha diversity of microbiome, means the heterogeneity of microbes within a tissue of study. So in this case, basically, what we find is that patients who live long term, they just have more heterogeneous microbiome, like different types of species within the tumor. This was described before as a kind of beneficial or positive predictive biomarker in the gut also. So in previous work looking at gut microbiome, higher diversity was also correlated with better outcomes. But we did find
similar outcomes, but this is in terms of tumor microbiome. So this is... Yeah. Have you looked at the microbiome of normal mice's pancreases versus, ones with tumors, and then tumor versus the rest of the pancreas? Have you looked at those comparisons?
Yeah, we did look at that, but the tumor microbiome of normal mice, I mean, you can amplify, but it doesn't, you don't have as many bacteria as when you have a tumor. Even the adjacent area of a tumor have different microbiome than the normal pancreas in complete absence of tumors. So, instead what we did is we did focus on the tumor microbiome itself. What you're asking is certainly, something that we are looking at right now, but we don't have conclusions about, normal versus normal adjacent, but yeah, we did focus the study on the tumor microbiome itself, mostly because those were the samples, on patients that we had, available. So,
yeah, I mean, there's a lot of questions here. If you look at metastases, and you compare their microbiomes versus their constituent organs.
Yeah,
I mean,
exactly.
There's that to look at. As the tumor grows, does the microbiome change? If the tumor is more heterogeneous versus homogeneous?
Correct.
I mean, there's just tons of things to look at, it sounds like.
Yes, yeah, there's been a couple of publications, one of them from George Miller, who looked at the sort of microbiome progression over time in mouse models in which, KRAS is activated. So, yeah, that's, and then the metastasis versus primary certainly it's an important question that should be answered. So, yeah, a lot of, very interesting questions are coming out.
Well, what's the goal here? So if you see that, a larger diversity of microbiome appears to correlate with better outcomes, why? Is that because no one bacteria is able to take a hold take hold and disturb the homeostatic state of the rest of the microbiome and the organ itself? Or what do you think is the reasoning behind that?
Well, I mean, the question about why diversity or high diversity is better, I don't think it's been answered so far, right? It's a concept, and most of the studies point in that direction, but I don't think we yet have any answer. I mean, I would just say, of course, if you have a predominant bacteria, therefore lower diversity, and that bacteria is sort of pathogenic, then you could explain why those patients will have worse outcomes. But it's hard to tell. I mean, there are people who is mostly looking at, KRAS really inducing like dysbiosis, and that dysbiosis sort of determining that diversity. Or there are other groups who are more, thinking that it's not the dysbiosis or the kind of, complexity of the microbiome, but it's rather the increase of, relative species, which may have a, sort of pathogenic role that matters. So I don't think we know, that has to be tested experimentally. And it's not, very easy to do.
Yeah, and then the bacteria, are they forming a biofilm? Is there any morphology to their presence around the tumor, and does that correlate with the morphology of the tumor?
Right. Yeah, well, that's a little bit different. I mean, the biofilms are easy to be seen, like within the colon, within, other type of more like flat surfaces. Within the tumors, it's a little bit different. It doesn't really have a clear biofilm, but rather it's sort of, heterogeneously dispersed. And in our previous studies, we did not really look. We did do a couple of stainings to, make sure the bacteria is present, and we also did grow, bacteria to make sure that, functionally we were able to rescue and, have viability. But we have not characterized the kind of physical, spatial location of the bacteria. That's something that we are doing, right now to understand, what areas of the tumors are the bacteria located, what cells are primarily, attaching to it or engulfing it.
Yeah, and then I guess tumor microenvironments, I guess, become anoxic or hypoxic as the tumor grows. I wonder if that selects for, anaerobic bacteria that are able to live near the tumor, and perhaps it's very different from the surrounding microbiomes. I don't know.
Yeah, those are great questions, and again, those harder questions to answer, from patient specimen, because we don't get, it's hard to get, different areas of the tumors, but from in murine studies that could actually be looked at. Yeah, very, 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 very good points. Yeah.
So what's your speculation from here? Like, where do you take the research, and what kind of answers are you hoping to get?
Yeah, well, I mean, there are basically 2 points here. On one hand, in our study, we did find that, besides the diversity finding, the patients who lived long term, they also had a signature of microbes in the tumor. And, from that perspective, our main question is, is this the egg or the chicken? So are these bacteria just a biomarker of, better, responses to, the treatment? All these patients had a resection. So, it's just a marker of better immune activation, or, have those bacteria been implicated actually in, on sort of anti-tumoral responses. So we are doing those studies now, trying to understand, are they just a biomarker, or do they actually have a functional importance in the pathogenesis of cancer? That is on one hand. And then on the other hand, we also want to sort of understand, if modulation of the tumor microbiome can also, change outcomes. The idea is, if throughout the progression of pancreatic cancer and likely other cancers, the gut microbiome gets disturbed, and through an axis, it sort of changes the tumor microbiome, and that ends up, suppressing the immune system. Can you actually, modulate that gut microbiome or normalize it with the idea that will normalize, the tumor microbiome, and, kind of switch from immunosuppression to a, immunoactivation state? Those are the 2 areas that we plan to sort of focus.
Yeah, I guess, well, I would think that there's probably communication going on between the microbial constituent and the cells that make up its host.
Yeah.
Maybe through exosomes, EVs, that kind of thing. And then the immune system itself, I mean, you're right, you don't know, depending on the microbial constituent of a given tumor, does that help it hide from the immune system? Does it do the opposite?
Correct.
Right.
Correct.
Can you intervene?
Yeah. Yeah. Yeah, and I mean, we are very interested on sort of those potential, right, as you mentioned, sort of intermediates, right? It might not ultimately be the bacteria itself, but it might be, like metabolites, secrete proteins that really are the ones that ultimately, have this effect on the immune system. So yeah, it's a complex process. It's not just the change of bacteria that it's going to make a difference. It's a lot more complex process.
Well, just the fact that a tumor has a different microbiome from its surroundings means that the bacteria are there because it's a preferential state for them. They're getting something from those cells. Those cells are different. The signature of those cells is producing a different microbiome that's attracted to those cells.
Right.
I mean, that's one way, I guess, to call out the fact that it's a tumor is to see that difference. But I guess it's a difficult puzzle to figure out.
Exactly. Yeah. What comes first? And it's the complexity, or is in the individual? Yeah, it's a lot of questions. But we're trying to structure the questions and try to answer in a very kind of stepwise way.
Well, I don't know if you're allowed to speculate, but what does your gut instinct tell you might be at play here?
Well, I like to have all the hypotheses and sort of experiment them in parallel. So I really don't think that it's going to be one or two bacteria that are going to be the relevant ones, just like the examples that we found on the long-term survivors. I think certainly the fact that diversity is across the both cohorts and in other tumors, it's important. Makes me think that it's a combination of things. On one hand, you may need that diversity of bacteria and that kind of heterogeneity of bacteria, so that there are few bacteria that are actually predominant, especially if those are pathogenic bacteria. And on the other hand, you may also need the presence of certain bacteria that may have a kind of immunoactivating property. So my gut feeling is that you may have to target sort of both diversity and also the specific good bacteria should be increased, and the specific sort of protomorgenic bacteria should be lowered. So it's a pretty complex process. It's hard to manipulate in that way the microbiome, but at least that's kind of my idea.
Well, it seems like the biggest way to manipulate the microbiome is to change the diet of the mouse or the person or whatever it is, and control their diet. I mean, if you have several different mouse cohorts with pancreatic cancer and you change their diets, I would figure that would be the way to affect it, most of all, if possible. Yeah.
So, I mean, those are things we have to test. I mean, primarily you could compare, the transfer of, normal microbiome from like another mouse that has no tumors, and then compare that with just the diet. I'm not sure if the diet alone will be enough to kind of reverse, the dysbiosis to like a normal state and keep it. And especially, in mice, we can actually do this early on, but, in patients, once the microbiome has been so altered, I'm not sure if the diet alone will be enough to, revert the dysbiotic, process and kind of normalize it and keep it normal. But those are all things that have to be, tested. They're very, very important questions. And then the question of how often we need to do this, because if you just normalize, that might not be enough, right? Especially if you still have the tumor. That might be a good strategy on patients that are going for surgery, for instance, in which you do that change prior to surgery, and then the idea is once you don't have the tumor, you should not have dysbiosis any further.
Right. You said way early on in the call that some people are at high risk for pancreatic cancer. What causes people to be high risk? What's been observed?
Yeah, so basically it's patients that either have some genetic mutations, like germline mutations that predispose them. They have basically higher susceptibility for certain cancers. For example, the BRCA1, BRCA2 patients, especially the BRCA2. Also patients with like STK11, this is called the Peutz-Jeghers syndrome, and others. These are genetic syndromes that basically predispose the patients to cancers, like in this case pancreatic cancer. That's one population, and then also patients with a strong family history for pancreatic cancer, like two first-degree relatives, like brother and father, or father and a first degree to them, like an uncle. So two relatives in the family, and likely puts the patient at also high risk for disease. So our idea is, we want to, this is not only happening here. There are several centers, in the states that are conducting screening on these high-risk populations. The idea, is to test patients that have, higher risk than general population, and, put them on a yearly screening program, and, basically long-term, we're trying to look at biomarkers, of disease, so that we can expand this potentially one day to general population from a very simple circulating marker, etc.
Right. What about people that have had pancreatitis? Does that predispose them?
Yeah. So definitely pancreatitis is a risk factor for pancreatic cancer. So patients with pancreatitis. In our institution, because it's a cancer center, most of our patients that had pancreatitis, for instance, they also have other of the risk factors. They either have a genetic mutation, or they also have a family history. But yes, I know of other places in which pancreatitis is a sole risk factor. And those patients are also screened annually. Also, new onset diabetes has been linked with pancreatic cancer early onset. So in other words, diabetes is also a risk factor for pancreatic cancer, but it can also be an early red flag. So we also add that criteria to our cohort. So patients that may have a mutation that elevates the risk, but not as much. If they also have new onset diabetes, then certainly they have a higher risk. So we also screen them.
Yeah, that's what I was going to ask you in a different way. What when someone has pancreatic cancer, what happens to the functionality of the pancreas? Do certain functions get compromised or amplified?
Yeah, exactly. So the two main functions. On one hand, you have the the release of enzymes to digest the food, and that certainly gets compromised. That's what most of patients are taking enzymes that helps them digest better the food. And then second, the sort of management of the glucose in the body, the redistribution, and kind of through the secretion of insulin. So this can also be altered, and as I just said, that's why new onset diabetes could be a red flag for early pancreatic cancer. Mm-hmm.
Oh, so someone with pancreatic cancer, I guess they would have a harder time controlling their blood sugar in certain cases.
Yes, exactly.
Okay, that makes sense. Yeah, I guess because it would impact the insulin production. Okay, or delivery.
Yeah, yeah.
What's when you learned that there was a microbiome associated with the pancreas and with tumors, like what were your thoughts? Were you very surprised, or how did you react to it?
Well, I think because we started by finding these cells initially, and our main question was, why do we have those cells there? That sort of triggered us to start looking at this. And then when we did find the bacteria, that was around the same time that Ravid found the bacteria on the pancreas too. And it was just, when we did find it by ourselves, you always wonder, is this a real finding? Because it was certainly surprising. We always look at the pancreas as a sort of sterile organ, but we did take in consideration that tumors certainly have a different physiology and physiopathology, so that they could have a microenvironment that is different than the normal pancreas. But then looking at Ravid's work, and then work from George Miller and others, really intensify that search, for like specific bacteria, within the human tumors. Yeah.
Are you working with any companies that have pancreatic organoids? I never heard of such a thing. I know they have like heart organoids and brain ones, but has anyone made a pancreas on a chip or a pancreas organoid?
So I'm not working with any company, but within a group at MD Anderson, we are looking at opportunities to start something like this. We have not done it yet, but that's kind of in the horizon. Yeah.
Well, it sounds like it'd be a good, collaboration, because you could say, all right, hey, these are the microbes we've seen. Maybe you want to somehow add that to your pancreatic organoid and let us know what you see, and vice versa. It would be a good collab, it sounds like.
Yes. No, certainly. Many things, come into our mind now after this study came out. So, yeah.
All I've done is point out like 100 more areas where you have work to do, it sounds like.
It's wonderful. We are very excited with this. We have a lot of different directions we are taking now. Yeah. And you've made all wonderful observations.
So with all these possibilities, like how do you decide what to target first? Like what have you decided is going to be the focus for the next, year or two? What do you want to figure out?
Well, so we kind of split the work between, trying to understand further mechanisms and delivery and how the, bacteria gets into the pancreas in the first place, etc., more like using, mouse models. But on the other hand, I don't want to delay, sort of potential translational implications. So on the other hand, we are looking into doing a very, short-term trial on patients in which, we will do a very simple change in the gut microbiome with the idea that, if pancreatic cancer patients have a dysbiotic gut microbiome, just by restoring normal microbiome, that should affect the tumor microbiome, and it should affect the, immune activation state of the tumor. So we will probably, do a few patients and compare, prior and posterior to the transplant, how do the tumors change. So it's basically like proof of concept, trial on patients just to see if only that change, does it change any signal in the tumor. And then we will build from there. We don't want to go to like large studies without, having that simple question answered.
Yeah, that makes sense.
Yeah.
All right. What's the best way for people to find out more and to follow the progress and, check in with your work in the lab and everything?
Well, we have MD Anderson has, website has, a lot of these embedded into, and we have a laboratory website also within MD Anderson site that usually tracks, the news in the lab and the publications, etc.
Okay. Well, very good. Anything else I should have asked you?
No, I think you asked a lot of great questions. It was really, really fun talking with you.
All right, Florencia. Well, thank you for coming. I really, really appreciate it.
Okay. Well, thank you so much.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.